Hey everybody, this is Michele. I am a PhD in cryptography and a faculty at Sorbonne.
And my name is Ann Kim. I have a background in computer science and molecular biology from MIT in both my undergraduate as well as graduate degrees. And now I'm a CEO and founder of a healthcare software company called Array Insights.
So this talk is about a
But we're
discussion.
also hackers.
This talk is about a discussion that we started having in Paris a couple of months ago, and we think is also particularly timing because just a couple of days ago, Vitalik really posted something that is very similar to what we're going to talk about today. It is about proof of personhood that is a tool that's really needed in the blockchain space right now. How can you prove without relying maybe on some sort of government tools or like any very really invasive tool that a person is a human and not a bot. And so there are companies that are thinking about how to do this properly. Some of them are using biometric data. And in the discussion with Anne, we try to think about what would happen if we were to try to use DNA. And during this discussion, we also realized that this tool could also be empowering in some sense in the way that they could also be used for researchers because the way that cryptography treats the data could allow for privacy, but at the same time could really help compressing the data since handling DNA data often incurs in long delays due to the large processing power needed.
At the same time, these tools can also be abused. And so we'll be talking about the societal implications and regulations and guardrails in place at the end of this talk.
Cool. So Anne will start maybe giving a little bit of an idea of the basics of bioinformatics that were required, at least for me, to learn about this topic.
Great. And then afterwards, Michele will use that foundation to show you how to do a zero knowledge proof on this type of DNA and show you a demo even. In the live version, I will be doing the demo, but this video, I guess, is on YouTube. And at the very end, we'll wrap up by talking about some possible scenarios for how this tool might be used and how might it be misused, and give some general frameworks and suggestions for the audience, and happy to take questions live. Great. So without further ado, let's learn about DNA. So bioinformatics is the use of genomic information and biological life sciences information for with your computer in order to do high throughput analysis. And so it all starts with this central dogma that we're all made of DNA. But what does that mean? You have this spaghetti called DNA, and you can split the spaghetti and it turns into RNA. And at the point where it's split, you can then have the RNA encode for proteins, and then these proteins make up things like the color of your hair or your waistline, etc. And so this is really important for how you look, but obviously for different diseases and how you might categorize or figure out what your ancestry is, which is the two kind of examples we use for the proofs ahead. So eventually, when you have this DNA, you have this protein, and how do you actually encode or use this? You have these letters. They represent single nucleotide polymorphisms, or SNPs. And so we use them in order to do analysis on whether you have a disease or not, or in aggregate, we're able to use it in order to do ancestry reporting. And so you have a DNA sample, which is made out of your spit. It turns into this raw sequence. Then you do an alignment of this sequence based on reference genomes. So not everyone has the same DNA, obviously. And so we have these reference human genomes that we use in order to say like where chromosome 1 starts on my genome and where it starts on Michele's genome. And then once we have that alignment, it's easy to do sort of variant calls or any kind of like parallel analysis of, oh, this spot is an A on Anne's genome, and this other spot corresponds on Michele's genome, and that's a C. And this means that I have darker hair than Michele, for example.
Mine are transparent.
So this is what a VCF file looks like. You have a column of chromosomes, the position on the chromosome, which is like relative and based on complications with reference genomes, as well as an RSID, which is more global in naming, and then what the allele typically is for people, but then what the alternative looks like. And you can look up frequencies for these alternatives online. It's pretty interesting to see all these tables. But what we are going to be focusing on are these first kind of 5-6 columns in order to do our analysis. So where do we get these VCF files? There are many different sources. The Human Genome Project and 1000 Genome Project are these international projects where they're sequencing a bunch of different people who have volunteered to have their data used for science, and then you can download it and respectfully, hopefully, use it for different types of analysis. And so that's where our VCF files come from in order to do these proofs.
Yeah, well, we're downloading all this data. We realize that often they are already in a FASTQ form, which is really not the processed VCF file that is already aligned with a reference genome. So there are other websites online that are pretty easy to find, or just with a Google search, you can find them where you can really download the processed file in form of VCF. You don't want to play too much with some tools and all these tools for bioinformatics that allow you to go from these raw files into the VCF format.
Yep. And so there are many different tools out there to do bioinformatics work. There's like BioStars and Plink, and the Broad Institute also has their own library called GATK. And so we tried using these libraries, but ultimately the simplest way for us to do analysis was using grep from the terminal. And so on the far left hand, you can see this like read from my terminal of simply just grepping for a position, but I got a ton of different results for just the position because it's relative to a particular chromosome. So you have a bunch of different examples of it from different positions. And so instead we used RSID. So this RSID we grepped, and then we were able to find out that in this particular VCF file, it has like a T, but it normally has like a C for that allele.
Right. So yeah, this is the alternate and this is the reference.
Yep. And the second proof point that we have, besides just identifying single nucleotide polymorphism, is also how to calculate ancestry. And so if you've ever done 23andMe or Ancestry.com, the way that those companies tell you your ancestry is they take 3 steps. First, they have labels of different people and their ancestry. Second, they cluster them in order to give an idea of what those similarities and distances are for those vectors. And then third, there's a projection. So it's really important to emphasize for the arithmetization process that Michele is going to explain for his proofs that at the end of the day, you have this genome, it's a string, and then when we put it into this ancestry calculation, it goes into a vector space where all these labels are clustered, and then the projection of where a new sample is in this clustering is going to give you a distance. And the calculation of the relative distance between different clusters is going to tell you your percent breakdown of how Korean, Italian, French, or Kenyan you are.
Yeah. So right now we're going to dive a little bit into the cryptographic aspect of this work that we did. And before starting with the hard cryptography, I wanted to give an idea of the scenario that I'm thinking about. What I'm thinking about is that there are, for instance, these companies that I just mentioned that are sequencing your DNA, and they are giving the VCF to you guys, to the person that has given their sample for sequencing, and this comes in the form, let's say, of a VCF file. And at the same time, they upload what I'm going to call the hash of the DNA, what more formally, technically, should be called a commitment to the DNA, into some sort of database. This database can be the blockchain, can also be like some governmental database, or any sort of database that can be considered to be accessed later by some verifier. And the idea is that at some later point in time, a verifier might want to understand whether this user has, let's say, a single gene mutation that is leading to a particular disease, or do some sort of racial profiling on this user, and they are going to use this commitment to the DNA and ask the user for, let's say, a way of convincing them that they have this single gene mutation. And the user can choose either to reveal the whole VCF file, which is going to be very expensive to check for the verifier because it has to go through the whole file, or they can also give a zero knowledge proof, which is going to, in a way, leak less information about the full DNA, in the sense that it's going to reveal just a little bit of information that they want, and is also going to be more efficient for the verifier. In addition to that, the verifier on their end will sort of get this DNA, get the zero knowledge proof, and then compare the two, and then check whether the proof is valid. In particular, right now, we're not even talking about, in this scenario that we have, we're not even dealing with the authenticity of the sequencing. So the hash of the DNA, is it done by a trusted source or not, or whether the VSCF file has been correctly sequenced. We're assuming that the sequencer is doing things properly, and even initializing maybe some parameters, and then like giving the rest to the database and the user. So I'm going to talk about these two cryptographic components in this scenario, which is the hash of the DNA and the zero knowledge proof itself. So what do I mean by the hash of the DNA? I mean, I'm talking about commitment schemes, which are a common tool in cryptography, and they have two fundamental properties. The first one is that they are binding. So once you give me the hash of the DNA, it is very difficult to find another DNA that has the same hash. In a way, they are collision resistant. And the second property is that they are hiding. It is not possible to retrieve any information about the DNA given its hash. Actually, the hiding property is sometimes considered optional. I didn't yet implement it in the tool that we're going to see later. And for some identification and re-identification purposes, this has to be sacrificed sometimes. But the fact that the hash of the DNA is going to be, for instance, in our case, constant size is going to be 256 bits, it is already going to hide a lot of information about the DNA, really, because information theoretically, it is not possible from these many bits to recover the full string of the DNA. And committing is really easy. A commitment scheme is essentially two algorithms, the setup algorithm that generates it in the public parameters that I'm thinking the sequencer will run it, and then the hash function that will sort of produce this little hash that is a compressed representation of the DNA. Now, with this hash, we might want to later do some zero knowledge proofs again, which are the steps that was the user with a verifier. And what I'm imagining, what is a zero knowledge proof? First of all, it's a cryptographic tool that allows us to prove that a property holds. So we have a statement and want to prove that this statement is true without revealing any further information beyond the fact that the statement is true. And there are three fundamental properties that they have. First of all, they are correct. So if the given property holds, then the whole proof should verify. Secondly, they are sound. So if the proof verifies, then the property must hold. And finally, they are zero knowledge. So no information beyond the truth of the statement is being revealed. And in the case of the DNA, what kind of property might we be interested in? Well, something that we might be interested in is checking a particular position of the DNA and see whether this particular position has, for instance, the base A. And so in addition to the algorithms for setting up and committing, we might have also a proving algorithm that takes as input the VCF file and takes as index I, and the base, let's call it A, that will sort of internally run and output a proof that will prove that at the ith position in the DNA, I have the base A. And by this position, I mean, it could be compared to the reference genome, so the relative position, or it could be also the RSID. I try to implement both, but I think it's worth noting that in terms of limitations, I can play fairly well with thousands, even probably millions of data. But after that, it will start becoming slow. The demo that we will do will be around 1,000 RSIDs, and it seems that that's more than sufficient for most applications. For law enforcement, it seems to be in the range of hundreds, and for the case of paternity test, it's in the range of 20 RSID that I have to look, 20 loci in the DNA that I have to look in order to be able to discriminate between individuals. And finally, I have the verification algorithm that will just take the hash, the proof, and the position and the claimed value the claim base, and will output true whether really in this hash at this position we had this base. But more in general, we can prove even more complex statements. One of them that Anna mentioned toward the end was we could also try to do inner product between different vectors of the DNA, which immediately will give us a notion of a distance. So we could check whether two DNAs are close or not. But really, we can do any NP-complete property. So I think one thing that we are really thinking about right now is what are statements that are really interesting, maybe besides ancestry profiling, racial profiling, or ancestry. And so how do these things work internally? It's pretty complicated, but I think there are two steps that are worth mentioning. The first one is that there is really this arithmetization component where we take this property that we want to test and we transform into some algebraic relation, let's say some properties between vectors or polynomials. For instance, just before when we were talking about racial profiling, we're immediately thinking about distance of vectors. And another example is that if in my DNA I have 4 possible bases and I'm encoding them as numbers like plus or minus 1 and plus or minus 2, then to check whether a DNA, a hash is really a valid hash of a DNA, I would have to check the following equation. No, I would have to check this quadratic equation in one. I would have to check that this particular polynomial is 0 in all the positions. And that's a valid way, for instance, of arithmetizing this statement that all the elements that have been hashed are these 4 encoded letters. And after we arithmetized this statement and made it like some algebraic relation, we use some cryptography, some public key cryptography, or sometimes even symmetric cryptography to transform these algebraic checks into checks that are done over algebraic objects. This was a big part of my PhD thesis. I'm very happy to talk more about this, but I think for now, that's more than enough to talk about. Yeah, I wanted to make a little demo and show like a little bit what the tool will look like, because I think we'll also give a clear example of what kind of speeds we're talking about. I just wanted to say that there are many commitment algorithms that can be used. I choose one that is particularly efficient because proofs are particularly small, so it privileges size. And because we have this sequencer that can do perhaps some parameter generation, we can also take advantage of the fact that we have a trusted third party that can set up some pre-computation for us. And so
for a sense of scale, some of the files that we're working on, these VCF files, are 32 gigabytes long. And so it's really important, especially when you're figuring out the distance, like having a very efficient system.
Right. So I'm here in this folder where I have an example VCF file, I guess with some RSIDs that are already flagged properly. And I have also a list of RSIDs that will be used by the program to sort of decide which elements are necessary to be committed to. And so I have this DNA executable that will do the generation of the initial parameters and will generate this file called pp.bin that will have like 45 kilobytes and is able to commit to at most 1,000 2 to the 10 different RSIDs. And so this is just to give you an idea of how big could be the public parameters. And yeah, and let's say that I want to hash a particular VCF file. This is the hash we're talking about. This is hex encoded, so it's 256 bits. So now I can also prove that in this particular VCF file at this position, I have a certain value. And this is the proof size. It's going to be 512 bits. So still pretty small. And this is really not dependent on how many RSIDs we're committing. They will always be this big. And the reason they are this small is because we're using computational assumptions. So we're able to say that it's really hard to find a collision or that it's really hard to forge a proof thanks to like some some number of theoretic problems that are really hard and they haven't been solved in a long time. In this case, the discrete log. And so I might also want to verify, and I forgot the syntax, so I want to verify that there's a particular index, this particular hash has this particular proof, and this proof is valid for the letter C, which is what I think was there. And it doesn't say anything because it's correct, but if I put something else, for instance, it will output an error saying that the proof didn't verify successfully. So yeah, this is an idea. We will be publishing the repo soon, and we will probably be adding something more about the distance in order to maybe test ancestry and some more fancy tools. And I am personally really interested also in hearing whether there are other applications or what kind of use cases could be useful even in a setting that is maybe outside the blockchain space, but also more in general, like in terms of research, because there are interesting properties here. Again, there is the property of privacy, and there is the property of compression. And with that, I'm going to go back to the slides.
I'm on mute. Great. So I think it's really important to always consider where these tools are being used and who will be using them in different scenarios where you could potentially protect yourself and protect others, and considerations for society at large. And so a lot of key players here. Government is the biggest, I would say. And this is just an example of some US regulations. There are obviously many more internationally regulations for protecting genomic information, but we're just going to talk about the Fourth Amendment and the Genetic Information Non-Discrimination Act, GINA, here. So the Fourth Amendment, I think everyone's pretty familiar with it in the physical space, that you are you have a right to not be unreasonably searched or seized, but genomic information is not here. And there's so many examples, especially in the GEDmatch case, where government is able to use genomic information because it's not necessarily under the purview of some disclosures of like the Fourth Amendment. So I think it's really important that although there are some protections because it's not super specified, the precedent of law means that you're not necessarily safe. The next one, GINA, is much genomic information. It even recognizes that there are benefits and also misuses for genomic information. And so this act explicitly wanted to protect people from any discrimination for employment and health insurance. But I think what's really important here is what it doesn't protect you from. And so there's clearly many different ways, one of which is like life insurance or anything else. And so I think it's really important that we keep in mind that the precedents of this law hasn't really been tested. And further, it's even just hard to prove in court discrimination based on like race or sex. And so because genomic information is even more invisible, I would say, or genomic discrimination is even more invisible than discrimination against things you can actually see, this is something that is pretty dubious to protect you in court. Next.
I would say additionally that as most biometric information, oftentimes when we think about the data held in a database, whether it's held securely or not, there is a problem with what happens in the future. And so I think for me it was particularly concerning also to think about what happens when this hash maybe goes into the wrong hands. Maybe you can do proofs about things or other people, and the DNA samples, you're spreading them everywhere, and you cannot change them. So that's something that I've been always kind of been worried in terms of like how will the regulation evolve over time.
Yeah, and with biometrics, it's even trickier because there are so many complexities and subtleties that science is continuing to extract from biometric information. In fact, a couple of years ago, maybe it was like 2018, I had a presentation at DEF CON called Selfie or Mugshot. And the idea was like some of your selfies, maybe in the future, in aggregate, along with some reference database from 23andMe plus Facebook, perhaps, could actually be used for extracting genomic information from simply a selfie. And so because from an information theoretic, like we can't necessarily predict what that looks like for vulnerabilities, but we have like a vague idea, I think generally it's really important to have a lot of discretion when you're thinking about what to reveal and share about your biometric self. And so in conclusion, the name of our talk was, can I put my DNA on the blockchain? In hindsight, we maybe would have wanted to change that title, but we conclude that no, your mom would not want you to put your DNA on the blockchain because there is access and vulnerabilities for genomic information that are irrevocable. And to Michele's point, like you don't know how future provers are going to be able to misuse this data and how different entities and stakeholders will be able to misuse it as well. Regulation exists to protect you, but proving abuse is really hard. I think that's so true for just so many other simple cases. And then to be safe, use your own discretion. Don't put your DNA where you don't want it.